We talked a little bit about access to therapeutics already, and I know you're passionate about it, and you mentioned that simplicity fuels access, which I agree with you. Simplifying the processes, especially for legacy biologics and well-characterized biologics, it helps and facilitates access. How else do you think now and in the future, what other leverage does technology provide in the access, in the strive for better access globally to
Right.
therapeutics?
So we talked about gene therapy earlier, right? I mean, can you reduce the cost of measuring a titer and by using one technique versus 5 techniques? Can you reduce the amount of sample that is used for assay development? Instead of 30%, you use 5% of the sample, so the rest can go to the patient, right? These are pretty important problems. Let's go to the other extreme and maybe take the example of biosimilars. Right? I mean, often you have to do bioequivalence studies to show equivalence between the originator and the biosimilar. And I know there are alternatives being proposed and thought through, but that's the state of affairs. And those studies can cost between 50 and 300 million, according to some McKinsey surveys that we saw. 50 to 300 million is a lot for a generics manufacturer, right? With the risk that you have that you might not show bioequivalence. So how can you translate that need into an in vitro measurement? I mean, at the end, you're looking at physics and chemistry, right? And ultimately, if you can measure the chemistry using HPLC with UV and HPLC with mass spec, and mass spec is the most exquisite technique to get chemical information. So you can break it down and reconstruct it and say, well, this is the chemistry that I have. This is the constituent species in your molecule. The thing that's left then is the physics, right? Can you marry that to hydrodynamic radius measurements, interactions using light scattering, right? So if you take LC, mass spec, and multi-angle light scattering, you have an armamentarium of techniques that allows you to get the biochemical and the biophysical information that should obviate the need for some of these trials or delay their need in case you see some untoward effects, right? Can we have that discussion? And that discussion is being had now with the regulators, right? It will require a collaboration to say, okay, Humira X and Humira Y have the same chemical and physical footprint as measured by LC and mass spec and multi-angle light scattering, all data submitted through Empower with the most robust data integrity possible. So you have no doubt that the data has not been adulterated. X is equal to Y physically and chemically. Can I assume that biologically they will be the same? And even if they're not, there is no harm done, right? But it has saved me 50 to 300 million. And instead of having one biosimilar, I have 15 biosimilars on the market, hence reducing the cost, increasing the access. And that to me is a significant advancement. Now, if you take that and extend it further, you could do that at each step in the process, right? And what that does is, if you can assure after each unit operation, upstream bio out of the bioreactor, after a filtration step, that molecule A is equal to molecule B with physical and chemical characterization, that allows you to continue to optimize the process, which is something that we can't do today, without having to refile the process, right? And that to me is such a relief. I mean, as an engineer, it is something that I've lived with for 25 years. I started my career as a vaccines engineer at Merck Sharp and Dome. And one of the first assignments I had was to go and examine the process of producing measles vaccines. Measles vaccines were produced in roller bottles using chick embryo cells, right? And that process is, I suspect, not well controlled. It is pretty natural. And my boss had asked me if I could come up with a set of process improvements or a new way to manufacture the process. After 9 hours in the GMP suite on day one, I came out very happy saying, yep, oh, this is straightforward. We should go to cell culture and we would do A, B, and C. And she said, not only can you not do that, you can only modify the process as the speed of the roller bottles, the transfection, etc., within small ranges of variables. Disappointing, something that a persistent engineer who studied engineering for 8 years, 4 years undergrad, 4 years in a PhD, coming out and saying, I can't apply any of my engineering to improve this process. Long story short, I spent a whole year arguing with the team, and they decided the best thing to do was to promote me out of the department. So I went on to small molecules, but I never forgot that experience. And I said, you know what? Got to figure out a way to separate the process from the product. And better analytical techniques, collaboration with regulators, collaboration with manufacturers will relieve us from being hostage to processes that we develop today, or having our future generations be hostages to lack of ability that we have today, right? And so I think that's something that's important for all of us. I know you get motivated by it, I get motivated by it, a whole bunch of us get motivated by it.
I think it's very interesting that you, I sort of went the opposite way. Well, I also started in biologics and then had to learn about small molecules later in my career, sort of similar journey. It's very interesting that you're basically positing that sec-mols will be as powerful for biologics as dissolution, in vitro dissolution science was for small molecule, if I understood you correctly, which I think is a very exciting prospect. I totally agree with you, and I think the idea of being able to apply that measurement across the development process, and being able to make those adjustments dynamically is very interesting. And I think it aligns with the regulatory evolution that the regulators are moving towards risk-based and data-based assessments, and as it should be. So
I totally agree. I mean, I think sorry to sort of interject. I totally agree. In fact, it's even more relevant for high volume peptides now with GLP-1s, right? We're working with the GLP-1 manufacturers, and they've adopted our patrol systems. This is the old HPLC system that measures at line, online, from different chromatography columns, and the aspiration or the ambition of the manufacturers is to submit that data to regulators through Empower again. And I keep using the word Empower. Empower is the software that the regulators trust, so that they believe that you've not adulterated the data. No analyst has gone in and pressed a backspace without anybody approving or knowing. And that's very important for them to have confidence in the data that's being submitted, but it's being done online. So you're pulling samples from four parallel, and I saw this in the manufacturing suite of a GLP-1 at one of the top customers we have. They pull samples at line. The sample goes to the patrol system, and there are four big patrol systems right outside the GMP suite. And they go in, the data comes out. You go to the workstation that's in a different room, and you're analyzing it online and optimizing the process. And that data can go to the regulator at line, so that when you come to QA/QC, the burden is much lower.
Yeah.
Right. That would save time. That would save cost. That would speed up the whole process, while giving you the same quality attributes. And I think that's a clear example of quality by design, and something that the petroleum industry has been doing for years. Right. I think we can start to adopt these, especially for high volume applications. And I know the regulators are super interested, and people are collaborating with them at this point.
That's very exciting. Of course, the next extension of that, especially since we talk about data, is the use of AI and machine learning. Where do you see the biggest impact from your vantage point for this technology, and how do you see that both enabling, but where do you also see potential challenges or concerns in terms of what the risks are in your space?
I think it's a fantastic question. One that I think if you're a leader today, if you're not wondering, you're not awake, right? I mean, this is something that you get confronted with almost every day. The tools, the power of using retrospective analysis on reams of data, or projecting forward based on the past using correlations. But I think it's worthwhile taking a step back and being clear on what we're trying to do, right? So machine learning, meaning retrospective examination of data and analysis, allowing you to come up with more intelligent predictions, is one approach. The other is using large language models to say what will happen if A, B, and C happen now, right? Those are two different things. We've used machine learning approaches, watching operators for hours and hours, recording what they've done, and applying that to, for instance, integration of peaks in HPLC, right? If you've ever done any experiment in a lab, right? It's HPLC or DSC or something, wherever you're integrating a peak, and we've all integrated peaks for different reasons. If you leave it up to the user where the starting point is and where the ending point is, even if you use sort of sophisticated analysis, second derivatives of inflection, you still have a judgment, right? What if you were able to look across every lab and use the judgment of every user and came up with an average, a median, a standard deviation, and agreed on what that standard is, and gave that to the user and said, this is the standard we will use. It will not be up to user X at 1 a.m. and user Y at 3 p.m. to determine what this impurity is. And you're able to integrate it, quantify it, and come back and agree on an answer based on an algorithm, right? Which has been trained by you and by millions of other analyses. I think that's a huge step forward. And we've already supplied that to many of our customers through the Alliance IS, right? And so that's a very practical example, right? And
yeah, I think for in that particular setting, it's hugely enabling. To me, it's the analytical. I always say it's the analytical equivalent of calling balls and strikes in baseball. You know, having that box that's been calibrated. And yeah, I completely agree with you.
And then spending your time on signal and not noise. Exactly. I mean, that's what we're trying to do, right? I mean, your time is valuable. You don't want to be spending time on noise, and you want to figure out which peak is a signal, take that species, put it on a mass spec, analyze the heck out of it, and tell people, is it good, bad, or indifferent for my sample? And you want to do that as fast as possible, right? So for me, to enable customers, to enable users to do it, we can use retrospective analysis, right? So that's just one example. The other examples are internal. We're using it for supply chain optimization. We're using it for understanding what are the order patterns for our customers. What are the replacement cycles for different types of instruments? How often do people replace them? Under what conditions do they replace them? What are the conditions that are sort of sufficient for us to go to a customer and say, hey, your service charges are actually very high. You might want to think about the next generation of X, Y, and Z instrument. So I think many different applications, but what we've done is been very deliberate about saying, okay, the tool is here. The teams sort of built a data lake. We have the master data cleaned up. Now tell us what applications you can conceive. So we had 50. And so what we did is we asked people, we had over 50 ideas that came through the organization. We picked 10 pilots. 8 out of 10 pilots seem to have yielded good results. 2 out of 10 we killed in the middle to say, okay, this is not achieving what we thought. And one of the most interesting ones that didn't succeed was there was a discussion on having ChatGPT rewrite code of old software.
Oh.
Right? So we said, okay, let's train it and let's try it out. And I'd heard this from my kids. They said, but you know what? I can use my computer science assignments using ChatGPT. Why do I need to learn these languages? And of course, one of them tried it with his friends, and the teacher found out, and he was in trouble. We got letters, and that was great that we did, because he was supposed to learn the language and not use ChatGPT. But that's an aside. That said, it's not as good. It doesn't work. It doesn't take empower and rewrite the code in any modern language, okay? So we still have to have human beings intellectually take a step back and say, this is what empower is trying to do, and this is what I need to write in the new code. So I'm sensationalizing for a reason, and I'm choosing extremes for a reason. It's not totally unsuccessful, but it is not successful to a point where you would take something as robust as an Empower software and move it into this new zone.
Yeah, no, I mean, that is comforting to hear, and it's a good segue to talk a little bit about leadership and human competencies. So, in your space and in your environment, what are the competencies that you look for the most in your direct reports?